So I want to take a minute and go ahead and focus on one particular group of proteins, and this group is the enzymes. They do some important things that I wanted to go ahead and highlight in terms of allowing chemical reactions to take place inside your body. And most of what happens inside our bodies is some higher level of chemistry if we go ahead and focus in on the smaller details of it. So let's go ahead and take a look at our essential questions. So, essential question number one, what group of macromolecules do enzymes belong to? Hopefully you can go ahead and answer this question already. It's one of the subgroups of proteins. Number two, what are enzymes needed? Why are enzymes needed in biological systems? And so we're going to talk very specifically about a function that they serve in allowing these chemical reactions to take place in our body. And then lastly, what are enzyme inhibitors? And we'll go over three types of different inhibitors and what impact these have on these super important enzymes. So first things first, enzymes are responsible for a lot of the key chemical reactions that take place in our bodies. Some of these chemical reactions need to have an activation energy that's just too high, or they take place too slowly. And so the enzyme's job is to go ahead and speed up that chemical reaction by lowering this activation energy to allow them to take place within practical time spans for our living tissues. So ultimately, if we define an enzyme, it's a protein that acts as a biological catalyst. That biological term, you're probably pretty easy to familiarize yourself with. Bio, life, this is a living catalyst. Okay, so then what is a catalyst? Well, a catalyst is a substance that speeds up the rate of a chemical reaction. We have in chemistry all sorts of catalysts that don't have to be alive. But inside our bodies, all of this is taken care of by these living catalysts that we call enzymes. So faster reactions with less energy requirements so that we can do more of them to carry out all the processes our body needs every single day. Okay. If you remember when we went over chemical reactions, we talked about the fact that we start with the reactants and end with the products, but that you always require a certain amount of activation energy in order for the chemical reaction to occur. If you take a look at this graph here, you'll notice the impact of the chemical reaction. Okay, now here are the reactants, here are the products. So if the reactants have a higher starting energy and a lower ending energy, this is an exergonic reaction, or a reaction that gives off energy. But you'll notice that here's the peak. We got to go up from here to here. You'll notice the arrow here. From here to here, that's the activation energy that that particular chemical reaction requires to start. However, when we put the enzyme into the area where the chemical reaction is occurring, notice what happens. We still require activation energy, but in this case, it's about half as much the activation energy as you would need for the same chemical reaction without the enzyme. Okay, the starting point, the starting energy for the reactants, and the ending position for the products is the same. Those parts do not change. You still start at the same place, end at the same place, but notice that your energy requirements for that activation energy are cut in almost half. This is the impact of an enzyme. It saves our body so much energy by doing this so that we can go ahead and carry out more chemical reactions far more efficiently. Now, when we go ahead and start talking about the parts of an enzyme catalyzed reaction, they change the names a little bit. The reactants that are involved in an enzyme catalyzed reaction are now known as the substrates. Okay, so in this particular instance, what we're looking at here, let's see here, is essentially the reactants. Okay, the reactants and the substrates are basically the same thing. The only difference is that this takes place in an enzyme catalyzed reaction. Okay, so enzyme catalyzed reaction. Specific substrates are going to attach to a a particular spot on the actual enzyme, and the spot on the enzyme where this reaction takes place is known as the active site. So these substrates, which remember are reactants in an enzyme catalyst reaction, attach to the active site, okay, and that's where the chemical reaction is going to take place, and that's why we have lower activation energy needs. So the enzyme and substrate are going to bind together, and then it helps with alignment to lower the activation energy needs. And so they do so through what are known as intermolecular forces, which you'll talk more about if you go on to take chemistry, to form what's known as an enzyme substrate complex. And they'll remain bound together in that form until the chemical reaction is done and you go ahead and produce your products. Now, the products, that part does not change, still called products when we're finished with the chemical reaction. So let's see this in action. So if we go ahead and take a look here, you've got your enzyme, and this particular enzyme we're looking at is hexokinase, and you can see here is that active site where we're going to go ahead and bind to the substrates. So our reactants in this case are glucose and ATP, but we're going to go ahead and call those, yes, substrates, because they're in an enzyme catalyzed reaction. So the substrates are going to go here and attach to the active site of the enzyme. And this is where the chemical reaction is going to occur. When they're attached together, the enzyme substrate complex is the whole thing attached together. That's going to cause the chemical reaction to take place, and that's going to release the products out. So the product cycle is pretty much the same as it was before. We're just changing the name of the reactants to substrates because of the enzyme, and those are going to attach to the active site to create the enzyme substrate complex. All right, so again, substrates, that's the name we're going to use for the reactants, but on the other end, we're still going to use the term products. So I have a short little video going over the role of enzymes, and then we'll go ahead and finish up.
At any moment in time, there are millions of chemical reactions going on at the molecular level in every cell. Virtually all of these reactions need catalysts to provide a site for the reaction and to coordinate the energy flow. The catalysts in cells are proteins called enzymes. The molecules entering into reactions are called substrates. Enzymes catalyze both endergonic and exergonic reactions. For a catalytic reaction to occur, a substrate must fit into a groove on the surface of the enzyme called the active site, so that many of its atoms nudge up against the atoms of the enzyme. Reorienting the molecules in this way reduces the amount of energy needed to activate the reaction. The fit between the enzyme and the substrate must be tight, like a foot in a tight-fitting shoe. Each cell contains from 1,000 to 4,000 different types of enzymes, each with a specific substrate that it can catalyze. In this process, enzymes are not used up or destroyed. They are merely intermediaries in the reaction, and after it is over, they are able to catalyze other reactions.
Okay, so a couple things to highlight it here. The enzymes are not used up in the chemical reaction. When the reaction is over, they can be reused again and again and again. However, each chemical reaction only has one specific type of enzyme, and that enzyme can only carry out one chemical reaction at a time. Okay, so enzymes can only carry out one type of chemical reaction and only one at a time, but when the reaction is over, the enzyme is freed up to go ahead and do the reaction again. Now, there are several factors that affect the rate at which enzymatic reactions will proceed. So how quickly can we go ahead and go through a chemical reaction? Well, the first thing that impacts it is temperature. Temperature causes molecules to move faster, therefore it's easier for them to reach the active site, and then carry out the chemical reaction. pH. pH impacts the functioning of the protein, or in this case, the enzyme. So the shape of the enzyme can change as the pH changes, and there's an optimal pH range for any enzyme. So enzyme concentration. Obviously, the higher the number of enzymes, the faster the chemical reaction is going to happen. Likewise, the more substrate you have, again, it's faster to reach the enzyme, so the more the chemical reaction will take place. However, there is a limit on this. You can get to the point where there's so much substrate that the enzyme can't pick it up and they end up blocking each other, and it can actually slow down the rate. And lastly, the presence of any inhibitors or activators. So activators actually make it easier for the chemical reaction to occur. Inhibitors actually slow down or stop the chemical reaction from happening. So let's focus a little bit on enzyme inhibitors. Now, enzyme inhibitors are substances which alter the catalytic reaction of the enzyme. So we're slowing down the rate, sometimes all the way to a stop. And there are three typical types of enzyme inhibitors that we typically see. One, we can look at competitive inhibition. Two, we can look at what's called non-competitive inhibition. And lastly, substrate inhibition. So let's start with competitive inhibition. In this particular case, you can see that we have a substrate right here that fits into the active site of the enzyme. But there is another substance that is similar in shape and fits into the active site, but does not allow the chemical reaction to occur. So this fits in really well. Chemical reaction happens, we make product. If this one ends up in the active site, the chemical reaction doesn't occur, and now the enzyme is bound up and can't do any other chemical reactions. This is what's known as competitive inhibition. In order for this protein to actually do the reaction again, we would have to remove the substance that's blocking it and allow the entry of the material we do want to happen. The next one is what's called non-competitive inhibition. In this particular case, the inhibitor is something unrelated to the substrate, but will bind to the enzyme, often in a place that's not next to the active site. And when it does, it changes the shape of the protein enough so that the active site changes shape, and the substrate will no longer bind to the enzyme, preventing the chemical reaction from taking place. So again, the substrate cannot reach the active site because the inhibitor attaches to the enzyme, creating a new shape that no longer allows the active site to grab a hold of the substrate. And that's known as non-competitive inhibition. And the last one is what we were talking a little bit with the rate, and that's what's called substrate inhibition. You just end up with so much substrate that it overwhelms the enzyme, and they're all kind of bumping into each other, and none of them are able to actually fit into the active site of the enzyme to allow the chemical reaction to occur. Okay, guys, that's all I've got for you. Thank you so much. I'll talk to you soon. So long.